The overall goal of this protocol for generating iPSC-derived human brain organoids is to model early human neurodevelopmental disorders.
This method can help answer key questions in the human brain development field, such as the role of centrosomes and cilia in normal neurogenesis and primary microcephaly. The main advantage of this technique is that it is a robust and quick method to obtain reproducible results from patients' iPSC-derived brain organoids. Demonstrating the procedure will be Elke Gabriel, a postdoc from my laboratory at the Center for Molecular Medicine, Cologne.
To begin this procedure, collect the neurospheres with a 200 microliter micropipette using a 2 mm tip previously cut with sterile scissors. Next, place the neurospheres approximately 5 mm away from each other on a paraffin film in a 100 mm dish, and carefully remove as much of the remaining medium as possible. Then, add a drop of EHS matrix onto each single neurosphere. Incubate the EHS matrix drops with the neurospheres for 15 minutes at 37 degrees Celsius. After that, wash the neurospheres carefully from the paraffin film by flushing them with neurosphere medium. Subsequently, incubate the neurospheres for the next 4 days and add 2 milliliters of fresh neurosphere medium on day 2.